We do, but I don't have that scheduled yet, so we'll get it scheduled soon. All right, guys, here's the first question we're going to work. A bag of M&Ms was purchased for a science experiment on probability. The teacher counted 100 M&Ms. The data are listed below. She hypothesizes that if there are 4 colors of M&Ms, there should be an equal number of each color in the bag. We want to know if her hypothesis is right. We're testing her hypothesis. So she counted out the M&Ms. And what she actually got was this. Observed. What we actually get, we're always going to write that down in observed. We're going to always make a chart like this. So she counted 18 brown, 22 yellow, 30 green, and 30 orange. Notice there's also a column for expected here. How did we come up with those numbers? What was her hypothesis? That if there's 4 colors, her hypothesis was that 1/4 ought to be each color, right? Well, how many did we count? How many M&Ms did she count? 100, so 1/4 of 100 is what? 25. That's where all those numbers came from. Yeah. No, we're going to work about 3 examples, so give me a minute and you'll see. But once you know the pattern of this table, calculating this statistic is easy. O minus E. That just means to take observed minus expected. I'm going to sit down here at the computer so I can fill this in for you as we go. So what's O minus E going to be for brown? Help me out.
18 minus 25.
-7, right? 18 minus 25. Exactly. So yellow's going to be -3, right? 22 minus 25. And that one's going to be +5 and +5. Make this column a little bigger so it makes sense to you. I'm trying to make those, that's O minus E squared. That's what that's supposed to be. So what's O minus E squared going to be for the first one here? O minus E is -7. This next column just means to what? Square that. So what's it going to be? 49. That one's going to be 9, right? -3 squared. 5 squared, 25. And 5 squared, 25. Over here, going to do the same kind of thing, except this time it's O minus E squared divided by E. So in this column, it's going to be 49 divided by 25, right? 49 divided by 25. 1.96. Let's just run them out to the hundreds place for right now. So 1.96. This one's going to be 9 divided by 25, right? Expected is 25. So 9 divided by 25. 0.36. And this one's going to be 25 divided by 25. Win. And the next one's going to be the same, right? 25 divided by 25. Now, to calculate what's called our chi-square statistic, what you see down here is the formula. The Greek letter chi sort of looks like an x. So it looks like x squared. It's chi-square is what we call it. What's this funny symbol right here? You should know this symbol from math. It's called sigma. What does it tell you to do in math? Sum up. Sum up. So that means take that column out of our table and do what to all the values in that column? Add them up. So we're going to take this whole column and we're going to add it up. So if we do that, 1.96 plus. That's not what I got. I got 4.32.
Oh, I was just ahead.
Yeah, so 4.32 is what that column adds up to be. That's called our chi-square value, 4.32. So let's put that in here. All right, now another thing you've got to know how to do is come up with your degrees of freedom. And if you read that part down at the bottom of the page, it sort of explains what it is. It's basically how many possible combinations there are. And what did they tell you, the degrees of freedom? How do you always find it?
One less than the total.
Right, one less than the total different outcomes you have. How many outcomes do we have here? We've got brown, yellow, green, or orange. So we got 4 outcomes, which means we have how many degrees of freedom? 3. Now there's a chart that you have. So first of all, we're going to put 3 right here. Then we're going to scroll on down just a little bit. This is a table of what are called the standard critical values. This table will always be provided for you. You just got to know how to use it. I'll explain in a minute why we always use the 5% column. But this is the column we're always going to use. 5% probability level, sort of the standard column to use. And 3 degrees of freedom. 3. So this value right here is the value we're looking for, right? 5%, 3 degrees of freedom. The value we get from this table is called our what? When you read through that, what did it tell you we're looking for from the table? It's called the critical value. It's what you get from the table, the critical value. So our critical value is 7.82. Now, in the chi-square statistic, what we're going to do is we're always going to compare our critical value, 7.82, to the value that we calculated for our data. And if we scroll back up, remember that our chi-square statistic was 4.32. I'm just going to call the critical value CV here. Here's the idea. If your chi-square value is less than your critical value, which ours is, if chi-square is less than the critical value, what do you think that tells us? When you read it, what did it tell you? If your chi-square value is less than your critical value, what does that tell you about the data?
The differences you observed and the expected data are, the differences between your observed and expected data are probably just interchecks.
Right. The differences between what we actually got and what we thought we should have gotten, very small. And probably just random, right? Who's the guy who did all the Punnett squares? The guy who figured out how genetics work. I know we haven't gotten there yet in this time. You guys should know that. Who's the guy who figured out genetics? Gregor? Help me out. Gregor, help me out. Mendel, right? Gregor Mendel. Say that, Colin. Gregor Mendel. He figured out, say if you bred 2 tall plants, and we'll get into the terminology in a minute, but let's say he did a Punnett square, and he figured out that 3 out of 4 ought to turn out to be tall. Well, when he actually bred those plants, did he get exactly 3 out of every 4 being tall? Nobody got what? Close to that. When you say 3/4 ought to be tall, that's a probability. If you take a quarter and you flip it, what do you know the odds are that you're going to get heads? 50-50. If you flip it 100 times, are you going to get exactly 50 heads and 50 tails? No, you're going to get really close to that probably, right? It's a probability thing. It's not perfect. This test lets us know is what we got close enough to what we thought we should have gotten that the differences are probably just due to what? Chance, luck. Now, on the other hand, let's say that our chi-square value, let's just say in this situation, instead of being 4.32, let's say we calculated and it actually came out to be 10.6. In that case, chi-square is bigger than the critical value. So what does that tell us? If chi-square is bigger than the critical value, what did your reading tell you had to do? Yeah, that your hypothesis is wrong. That the differences between your observed data and your expected data are too what? Too big. They're probably not due to chance. Your hypothesis is probably wrong, or maybe in the lab you just did something wrong, right? But there's a problem. So what do you need to do? If you're the scientist doing this experiment, what do? You redo it, and probably you're going to have to go back and change what? Probably going to have to change your hypothesis. But first, you probably ought to go back and redo it just to make sure you didn't do something wrong the first time. I'm going to pause our recording here for just a second. I'm going to show you a little video clip that goes through some of this. And then I'm going to do some more explaining and work. Okay, so here's sample problem number 2. And this is going to be just like the lab we did. This is what you're going to do with your lab data. A student completed a mitosis lab like the one our AP class just completed. The student counted 100 onion root tip cells and categorized them as being in either interphase or mitosis. I've got a typo there. The student hypothesized that onion root tip cells, unlike other onion cells, spend only 70% of their life cycle in interphase. Calculate chi-square for the lab and determine whether or not the data support the student's hypothesis. Okay, now the student counted 100 cells and just categorized in interphase or mitosis. Didn't break it up into prophase, metaphase, anaphase, telophase. Just interphase versus mitosis. And here's what they actually got when they counted them. Now that student's hypothesis was that 70% of the cells should be in what? Interphase, and 30% should be in mitosis. So how are we going to get these expected numbers? How many total cells are we working with here? Look at the chart. How many total cells are we working with? 200. So to get the expected number of interphase cells, what are we going to do? 200 times what? What was the hypothesis? 70%, right? Student hypothesized that 70% ought to be in interphase. So let's take 200, our total number of cells, times 0.7, 70%. When we do that, what is 70% of 200, guys? You don't really need a calculator for that. It's 140. So let's fill in 140 here. If we expect 140 here, how many do we expect there? 60, right? Now, do those numbers look pretty close to you? Do the observed and expected numbers look pretty close? Yeah. So probably what are we going to find here? Yeah, that the differences are probably just due to what? Chance. But always calculate it out. Don't just look and say, yeah, those are close. Because what looks close to you may not really be close. Let's calculate it out. O minus E. 158 minus 140. Going to get 18, right? 42 minus 60 is going to give us what? Negative 18. Now we got to go O minus E squared. So 18 squared, 324. And down here, that's just going to be 324 also, right? Because it's a negative 18 squared. And then this column, O minus E squared divided by E. So it's going to be 324 divided by, in the first row, 140, right? 324 divided by 140. So that's 2.31. Now on the second row, it's going to be 324 divided by what? By what? So we go. Second row, 324 divided by 60. And that's, having trouble with my calculator here, 5.4. So to find chi-square, what do? We add up that column. We sum that column. So that's going to be 7.71, right? Now let's scroll on now. How many degrees of freedom here? We had 2 outcomes, interphase or mitosis. So we have 1 degree of freedom. Scroll on down. Our critical value we have to get from that table, right? You've got the table in front of you, but here's the table. We're always using the 0.05 level, 1 degree of freedom. So our critical value is 3.84. Now, remember, we got to compare those. Our chi-square value is really bigger than our critical value. What does that mean? We have to reject our hypothesis. The student's hypothesis is not right. According to this data, is an onion cell spending 70% of its time in interphase and 30% in mitosis? No. And I showed you that intentionally because at first look, those numbers look pretty close together, right? You got to calculate it out though. Here it's saying that those numbers are too different for the differences just to be due to what? To chance. Something's wrong with our hypothesis, which means we have to go back and refine our hypothesis, right? Change it a little bit. So in this case, the answer to this bottom question, does the chi-square value support or reject the hypothesis? Reject, because our chi-square value is what? Greater than our critical value. Everybody with me on that? All right, let's work one more example. Yeah.
Say that your price was like 722 and the chi-square value that was like 1.04, would it still be acceptable because it's still lower, but it's like contrasting?
Yeah, anytime chi-square is less than the critical value, that means your differences between observed and expected are small and just due to chance. The only time you need to worry about rejecting your hypothesis is if the chi-square is bigger than the critical value. All right, now I mentioned Punnett squares a second ago. Here's the way we might use this with the Punnett square. Gregor Mendel bred 2 heterozygous tall pea plants. The plants produced 100 offspring. Of the offspring, 80 were tall and 20 were short. Draw a Punnett square and form a hypothesis. Test this hypothesis by doing a chi-square analysis. Now, I'm going to work this over here on the board. We haven't done anything with genetics much in here, other than mentions here and there. What does it mean to be heterozygous? What's hetero mean? Different. If a pea plant is tall but heterozygous, that means it probably has one gene that says be tall and another gene that says what? Be short. You've all seen this in regular biology. So that's what it means to be heterozygous and tall. Tall being what kind of trait? Dominant, and short being recessive. So the problem is saying we breed 2 of those, big T, little t organisms. Now I know that from regular biology you did Punnett squares between this kind of thing. During meiosis, what happens to these 2 T's? Right now this cell's diploid, right? What do we know meiosis does to diploid cells?
Right.
Makes them haploid. So it halves these, right? Some of this, let's say this is the father over here. Some of the sperm get the big T. Some of the sperm get what? The little t. Same thing happens in the mother. Those get halved. Some of the eggs get big T's. Some of the eggs get little T's. So let's fill this square in. These babies are going to be what kind of babies? Tall. Homozygous tall, but tall, right? These are going to be big T here, little t here. These are also going to be tall. These are going to be what? Tall. But these over here only get 2 little t's, so they're going to be what? Short. So my Punnett square is telling me we should get what? 3 tall to every 1 short. Those are what kind of values? Expected. That's what we expect to get. 3 to 1 ratio, right? 3 and 1. 3 tall to 1 short. So let's look at what we need to do here. Let's see if I put a table in here. Yeah. So we counted. Did I not change the numbers in here? Oh, yeah. Let's change that. Yeah. Plant height. There we go. So we counted 100 peas, and it told us that we actually got 80 tall and 20 short in the problem. Those are our observed numbers, right? 80 and 20. But according to our Punnett square, out of 100, how many ought to be tall? According to our square, out of 100, how many ought to be tall? 3/4 ought to be tall. What's 3/4 of 100? 75, right? Everybody with me on how I got that? The square says 3/4 ought to be tall. 75 out of 100, which means this has got to be what? 25. Now we just fill in our table. So 80 minus 75, 5. 20 minus 25, -5. And now we square those. So 25 and 25. And in this column, that first one's going to be 25 divided by what? 75. And that's the third. We didn't need a calculator for that. And this bottom one's going to be 25 divided by 25. Right. So our chi-square value is going to be what? 1.33. We add them up. And our degrees of freedom for this particular experiment is what? 1, because we had 2 outcomes. It's always 1 less than our total number of outcomes. So 1 degree of freedom. Now we go down to our table and we find our critical value. What's it going to be for this one? 3.84, 0.05, 1 degree of freedom. 3.84. Scroll on down a little bit. Well, actually, while we're looking at it, let's compare them. Chi-square is less than the critical value. Whenever we get that, what does that tell us? Our differences between observed and expected are small, and probably just because of chance. So what do we do with our hypothesis here? We're going to accept our hypothesis. Our hypothesis is essentially that 3/4 should have been tall and 1/4 should have been what? Short. We can accept that hypothesis because our data support that hypothesis. Does that make sense? All right, let's talk about what you're going to do.